Good afternoon, ladies and gentlemen. My name is Katharina Koetting. I work for the company Omnia, where I manage as the Director of Innovation and Technical Marketing our new product developments for agriculture. It is a great pleasure for me to be here today and to present to you the importance of two essential elements, calcium and magnesium, and their effect on soil health and crop nutrition. Calcium and magnesium are both essential nutrients which belong, besides nitrogen and potassium, to the most abundant nutrients found in plants. On the other hand, they also play an important role in the soil, where calcium is the 5th and magnesium the 8th most abundant element in the Earth's crust. What makes both of these elements so important for soils and crops, I would like to talk to you about in the next half an hour. The soil we live on and we grow our crops on are essential for life. Specifically, the upper few centimeters of soil layer are the important ones for food production. Keeping our soils healthy to produce healthy and sufficient food becomes more and more important in a world with a growing population and the expectation to grow more food on the same area of agricultural land. This is a challenge we are facing globally, and the focus on soil health is an important factor to reach these goals. If we look at the soil map, for example for South Africa, we see on the soil pH map one parameter that can demonstrate in which state our soils are. From the map, it is evident that the soil is not everywhere in a good shape. In the east, here marked in red, we see areas with acidic soil that go along with a low pH, which is often associated with an increased concentration of toxic elements, such as aluminum and manganese, for example, and nutritional deficiencies and other restrictive conditions for plant growth. While from the middle towards the west of South Africa, the pH is above neutral, which can indicate saline as well as sodic soils. Especially in sodic soils with a high salt concentration, the soil is poorly structured, and the high salt concentration may adversely affect crop growth. In order to keep soils healthy, it is important to keep the pH in a neutral to slightly acidic range. The reasons for low and high pH soils are various. Specifically, soil acidification is on the one hand triggered by natural processes, like nutrient uptake, for example, and mineralization of organic matter. On the other hand, it is accelerated by agronomic practices. One very important point is the application of acidifying fertilizers, such as ammonium salts and urea. As the ammonium nitrogen in fertilizers undergoes nitrification, meaning the conversion of ammonium to nitrate by bacteria in the soil, hydrogen is released, which leads to an increase in acidity. So every time these fertilizers are applied, the soil is more and more acidified, to which extent you can see here in the table shown on the right. The effect of such fertilizer inputs was demonstrated nicely in a recently published long-term trial from France. The trial was initiated in 1929 and had the objective to study the effect of fertilizers on soil properties in the absence of plants. The soil that they used was a so-called löss, a sandy clay loam texture, and it had an initial pH of 6.3. Over time, the control that you see here, without any treatment, showed a pH reduction to 5.6, going along with the decrease of soil structure. You can see this on the picture. This indicates the natural or environmental acidifying effects. Plots treated with ammonium nitrate fertilizer on the right showed an even more pronounced decrease in soil pH, down to pH 4.5, and also a very poor soil structure. The continuously lined plot, on the other hand, and here on the left, shows a high pH of 8.2 with a very good soil structure. These trials clearly show that the inputs of fertilizers do do not only affect the soil pH, but also soil structure. A consequence of poor soil structure is the slow wettability of the soil and lower water holding capacity. The long-term trials showed that the acidic soils, here in light blue, the lower line, needed more time for re-wetting than neutral plots shown in green, but also the overall water holding capacity is lower than for the neutral ones, which are here in the green line. So about 15% lower water holding capacity was observed in acidic soils compared to neutral soils. The reason for the compromised soil structure I want to outline on this slide. It basically lies in the cations that we find in the soil, their charge and the size of these cations. Calcium and magnesium both carry 2 positive charges and have a relatively small radius. This means they can hold clay particles in the soil perfectly together. They have a so-called good flocculation power, they are good flocculators. On the other hand, protons in the soil, in the acidic soil, and also the abundance of cations such as sodium and potassium are bad flocculators. Sodium and potassium both have only 1 charge, and the size of the molecule is too big to hold the clay particles together. Thus, the clay particles are far away from each other, cannot be held together, and the soil gets dispersed. So in order to improve soil structure, the application of calcium or magnesium is important. This can be either done in acidic soils, for example, with adding liming material, or in soils with a high pH by the addition of gypsum, for example. This improves the calcium and magnesium content in the soil and therefore the flocculation, which has a positive effect on water uptake, as we have seen, but also on soil aeration and consequently on the development of roots. The right balance of the cations is important for a well-structured soil and an efficient nutrient supply to the crops. A properly balanced soil contains around 80% calcium in order to maintain a good soil structure and an adequate supply of magnesium and potassium cations for crop nutrition. From the physical aspects of the soil structure, I want to move now to the chemical aspects. We have seen that the fertilizer inputs have a direct effect on pH. Furthermore, it was shown in this study, and it is known that soil pH is positively correlated to the CEC, the cation exchange capacity of the soil. This means the higher the soil pH was measured, the higher the CEC was. So keeping a soil pH at neutral range, the CEC in the soil can be increased, and therefore it has a higher capacity to bind nutrients and is thus more fertile. Additionally, the pH of the soil has an effect on the concentration of harmful cations such as heavy metals, manganese and alumina. Calcium and magnesium that bind to the clay-humic complex displace not only protons to increase pH, but they also displace other cations such as cadmium, for example. With an increase of soil pH and an increase of calcium in the soil, less soluble cadmium is available in the soil. This is shown on the left graph. This also has a positive effect on the uptake of cadmium by the plant, as shown on the right graph, which is shown to be reduced at neutral pH, leading to harvest with lower cadmium contamination, which is important, for example, for cocoa production and for chocolate. As already mentioned, soil pH and calcium concentration in the soil also has an effect on manganese. Excessive manganese in the soil can prevent the uptake and translocation of other essential elements like calcium, magnesium, iron, and phosphorus, for example. It also inhibits chlorophyll biosynthesis, which causes a decline in the photosynthetic rate. It was shown in studies that in soil with a low pH and a high manganese concentration, chlorophyll rates in the leaves are 4 to 5 times less compared to soil with a neutral pH and a lower available manganese. Thus, low pH can lead to high manganese levels, resulting in manganese toxicity symptoms. These symptoms vary widely among crop species, ranging from chlorosis to necrotic brown spots on leaves, petioles, and stem, or so-called crinkled leaves. The manganese toxicity shows another important effect of soil pH, which is on the bioavailability of nutrients. It is well known that the nutrient bioavailability is pH dependent and that an optimal bioavailability is observed at a pH between 6 and 7. For many primary and secondary nutrients, availability is increasing up to a pH of around 7, maybe 7.5. For some micronutrients, however, the bioavailability is reduced at higher pHs, specifically at pHs above 7. One example is manganese, that we have just talked about, that has a maximum availability in a pH range of 5 to 5.5, which is decreasing with higher pHs. Also, the availability of the macronutrients NPK is pH dependent, which is influencing the efficiency of the applied fertilizers. At low pH, NPK fertilizers applied to the soil cannot be converted and taken up efficiently and get lost. This was shown in a variety of studies, and from this we can deduce that only at a pH of 6.5 and higher, full use of the applied fertilizers can be made. This is an especially important aspect considering the currently very high fertilizer prices, with tripling prices for fertilizer input over the last 1.5 years. Only if the soil pH is right, full use of the applied fertilizers can be made. So it's recommended to regularly check your soil pH and get it right before NPK application. After looking at the physical and chemical properties which are affected by soil pH, I want to look also quickly to the effect it has on microbial properties. It was shown that microbial populations in general benefit from a higher soil pH. And I want to give you two examples. First, one for earthworms, you see on the left. They are much more abundant in soil with slightly acidic pH compared to acidic soil. And this, of course, has then a positive effect on the soil structure as well if you have a higher quantity of earthworms in the soil. A second example you see here on the right, which is quite striking for nitrifying bacteria. So these rhizobia that live in symbiosis with legumes, they fix nitrogen from the atmosphere. And we can see that only changing pH from 6 to 7 increases the number of bacteria in the soil significantly. So much more nitrogen can be assimilated from the atmosphere and supplied to the plant and in a neutral soil pH. How important the soil pH is for crop productivity was recently demonstrated in a Swedish study, which evaluated data from a national soil inventory and from Swedish long-term fertility experiments. The aim was to identify the most yield-limiting soil parameters and best agronomic practices in order to further improve yield. Among all the manageable soil parameters, pH was found to have the greatest potential to affect crop yield, being more important than factors such as soil organic matter or plant available phosphorus, for example. Plotting crop yields against pH values showed a strong correlation between these two parameters in the range of 5.5 to 7.2 for most crops, which you can see here on the right. Comparing the yield from fields with pH 6 and 7 revealed that the yield can be almost doubled for winter wheat and spring barley. Even at pH values of above 6.5, yields of cereals still increased significantly. Only at pH values around 7.2, cereal yields declined, which was attributed to a lower plant availability of micronutrients. So in contrast to the currently existing soil pH recommendations in Sweden that are more around 6, 6.5, a big potential could be shown when increasing the recommendations to a target pH in water of around 7. The practice of liming to increase soil pH is actually really an ancient agronomical practice. But in the past years, the practice of liming was more and more improved with the use of more efficient products and precise application techniques, such as variable rate application. One way to produce more efficient liming material is the use of ultrafine particles. I want to show a recent study here from Southeast Australia which underlines this effect. They studied the performance of limestone of different particle sizes, between several millimeters down to low micron levels, in a soil with a pH of 5.1 at the start. The ultrafine limestones with particles below 75 microns, here shown with the line with squares, showed a fast increase immediately after application, and it outperformed coarser limestone treatments clearly over the first 3 years after application, and still performed best after 6 and 7 years. Lower particle size means higher reactivity, therefore a higher rise of pH and more efficient liming products. Additionally, what they could show in this study is that finer particles can move deeper into the soil profile. They've checked soil depths of 10 to 15 centimeters, and while over the time hardly any effect was seen by the coarse material, the fine material could increase with time also deeper soil layers. So the fineness of the particles is very important for its reactivity. And this is due to the higher surface area that we have in fine particles, so more area for the reaction to take place. You can see this if you put a fine limestone, for example, in vinegar, as you see down here. It will react immediately and bubbles will form immediately. On the other hand, if you add a coarser limestone to vinegar, you will see no reaction. The same actually takes place in the soil, and it is especially important the higher the starting pH of your soil is. If you want to shift soil pH from 6 to 6.5, you need a much more reactive liming material than if you want to change the soil pH from 4 to 4.5, for example. This is shown here on the slide. The more acidic the soil, the easier the liming material can dissolve. For an example, you can see this on the picture at the bottom. For a coarse limestone, if you put this in HCl with a low pH, you will see immediately a reaction, while if you put the same material into water at a higher pH, no reaction will take place. So the reactivity of the liming material is pH dependent. In very acidic environment, a strong reaction can be seen and a fast reaction will take place, while at a higher pH, you require a much more reactive material in order to still increase pH to higher levels. Another important aspect is the dispersion of particles in the soil. If you have very fine particles, you apply the same dosage, you will apply more particles onto the soil, and therefore you have a bigger surface area where the neutralization reaction takes place. And this neutralization reaction only takes place really around the particle in a few millimeters. So if you have only a few particles in your soil, only a small soil volume will get neutralized. While if you have very fine particles and therefore many particles with a high surface area in the soil, around these particles the reaction will take place and you will neutralize the soil more homogeneously. Even though the importance of a correct soil pH is well known and was demonstrated in many studies, and it is moreover well known how to improve soil pH, we see that liming is still not practiced comprehensively. To better understand the status of soil pH and the practice of liming in Germany, a study analyzing the pH of cropland and grassland based on around 3,000 soil samples was performed recently. A map was created based on this analysis that reveals low soil pH, especially in the north of Germany. Overall, the study has identified that the soil pH of 52% of the grassland and around 41% of the cropland are below the current pH recommendations. meaning there's still a lot of potential to further improve productivity in Germany by a more regular practice of liming to optimal soil pH levels. I want to summarize the key messages for this first part of my talk. First of all, calcium and magnesium are very good flocculators, so they're important to create a good structured soil. Furthermore, soil pH has effects on the physical parameters, on chemical parameters, but also on biological soil parameters. So it's much more than just soil pH. Large-scale studies that I've shown you have illustrated how important the correct pH management is for crop productivity, and even much higher pH recommendations than we are currently using given optimal yield. And last, the high reactivity of soil amendments is really important to result in a fast but also long-lasting pH adjustment to optimal pH levels. In the second part, I want to talk more about the nutritional aspects of calcium and magnesium. But first of all, I want to introduce a bit to the nutrient transport system in order to understand how calcium and magnesium are transported in the plant. So we have two transport systems, the xylem and the phloem. In the xylem, water and all minerals are transported in one direction, from the roots up towards the leaves. It's a so-called unidirectional transport that is driven by root pressure and by transpiration and only goes upwards. In contrary, the phloem transports nutrients like sugars, but also some of the minerals from so-called source tissues to sink tissues. A source tissue is a tissue that is performing photosynthesis and accumulating sugars. And sink tissues can be on the one hand the root, so the transport goes downwards to the root. But sink tissues are also newly developing leaves, fruits, or flowers. So this transport is so-called bidirectional. What is important to know is that the flowability or the mobility of nutrients in the phloem is very different. And calcium, also manganese, have a very low phloem mobility. This means they can only transport it in the xylem, or mainly transported in the xylem, from the root towards the shoot. If you apply calcium to a leaf, you will hardly see any translocation to the root or to developing leaves, in contrary to phosphorus, for example, which is very mobile. If you apply it to a leaf, you will see it throughout the plant. With calcium, this is not possible. It's basically transported from the roots to the leaves, and you need to, it's taken up at the time that it's needed. It cannot be stored and retranslocated anymore. And this is really important to understand calcium nutrition and to understand how to, and at which time points to apply calcium for the crop. So what are the functions of calcium in the plant? Calcium is very important to maintain the structural integrity of plant cells, as it is part of the cell wall and the cell membranes. Moreover, calcium improves the quality and storability of fruits. It promotes the expansion of cell wall, which is very important, for example, in root hairs and the pollen tube. Additionally, it helps to maintain the cation-anion balance in the cell, and it is functioning as a secondary messenger. For crop production, the function of calcium in the cell wall and cell membranes is very important. Here it gives stability and strength to the plant. On the one hand, calcium is part of the cell wall, which you can see here in the graphic on the top, where the positive charges of the calcium molecule bind together the pectins from the cell wall, which are negatively charged. As well, the similar function calcium has in the biomembranes, where the positive calcium binds to the negatively charged phospholipids to keep the stability of the membranes. So this stabilizing function is very important to keep plant cells intact and to give strength to the plant. If we don't have enough calcium, we observe calcium deficiencies, and this is mainly found in acid soils and in horticultural crops which have a high calcium demand. It occurs if there is no adequate supply of calcium. For example, in low or not transpiring tissues, I told you it's transported with the xylem with the transpiration stream. So if you have low transpiration, you usually also see a low accumulation of calcium. And it cannot be remobilized really from older tissues to developing tissues. So a too low concentration of calcium leads to a breakdown of cell walls and then a resulting cell death. Typical calcium deficiencies you can see here on this slide. So it's especially in young expanding leaves. In lettuce, for example, we call this tip burn. So these leaves have a low transpiration, therefore low calcium accumulation. Same is true for celery. There the inner leaves have a low transpiration, therefore low calcium supply. And here the deficiency is called black heart. But the same is also true for so-called blossom end rot in tomatoes, pepper, and melons, which have if they have a low calcium concentration, the rotting starts from the blossom end. Other deficiencies are so-called bitter pit in apple, or also fruit cracking that we see in cherries or tomatoes. One of the functions of calcium is the strengthening of plants, which also has a positive effect on the reduction of pathogen attacks. As calcium gives stability to the biomembranes, lower amounts of plant exudates can be released, thus less infections are triggered. Likewise, calcium improves the stability of the cell wall, which inhibits the pathogen attack of pathogenic enzymes to dissolve the cell wall, and it makes it more difficult for the pathogen to infect the plant. I want to give you an example on the right for botrytis, but the same can be seen for many other pathogens, fungal but also bacterial pathogens. Here it is shown that the infection with botrytis is reduced with a higher concentration of calcium in the plant. It is not related to the concentration of potassium or magnesium, but only correlated to calcium. The higher the calcium level is, the lower the infection with botrytis. Moreover, a good calcium nutrition is improving the storability of fruits. So if calcium treatment is done before harvest, the susceptibility, so storage diseases, is decreased, which we can see here in the example of apple. So the higher the calcium concentration in the apple is, the lower the percentage of rotting that is observed during storage. The key messages for calcium I want to give you is that calcium is mainly transported in the xylem with water, with a transpiration stream, and is very little mobile in the plant. It is an important building block of the plant's cell wall and membrane, giving strength to the crop. Calcium deficiency symptoms therefore only first appear in tissues with low transpiration, as it is transported with the transpiration stream. And thus you need to apply calcium when and where it's needed. It's not possible for the plant to store calcium and reuse it. Now I still want to take a look into magnesium and magnesium nutrition. Magnesium in the plant has several functions. A very important one is that it is a constituent of the chlorophyll molecule. And moreover, it activates several enzymes in the plant. For example, enzymes involved in CO2 assimilation, enzymes involved in carbohydrate transport, but also involved in protein biosynthesis. Just to illustrate again the very important function as part of the chlorophyll molecule. So you see here depicted the chlorophyll molecule on the right side. And really in the center of this molecule we see magnesium. This cannot be exchanged by any other ion, and it is essential for the function of chlorophyll. If we don't have enough magnesium in the plant, there is reduced amounts of chlorophyll, and this then can be seen as magnesium deficiency. Here you see such examples of magnesium deficiency. So we see that there's a low amount of chlorophyll in the leaves. They turn pale, yellowish, and this is especially visible around the veins, and therefore this effect is called intraveinal chlorosis. We see this in grapes, maize, potatoes, but also many other crops. The deficiency symptoms are very similar. Moreover, the deficiency of magnesium results in a reduced shoot and root growth. You see this here depicted in several examples. If there's an adequate magnesium supply, you have a proper shoot growth, a proper root growth. In conditions with too low magnesium, both of these are reduced. For an adequate magnesium supply, not only the amount of exchangeable magnesium is relevant, but also the ratio of other cations in the soil must be favorable for plant uptake. Magnesium uptake can be blocked by cations such as potassium, ammonium, calcium, or manganese, as they compete for the same transporters that facilitate nutrient uptake. Especially potassium can inhibit magnesium uptake, which is called potassium-magnesium antagonism. An adequate supply of magnesium is recommended in order to prevent this antagonism, increase magnesium nutrition, and thus crop yield. The importance of magnesium fertilization for crops was demonstrated in a meta-study performed in 2020, in which more than 300 datasets from China and more than 150 datasets from other countries were evaluated. The aim was to analyze how soil application of magnesium fertilizers affect crop production in the field. It was shown that fertilization with magnesium resulted in an average yield increase of 8.5% under various field conditions. Moreover, the ergonomic efficiency of the magnesium fertilizers was evaluated. The ergonomic efficiency is an important parameter indicating relative fertilization efficiency in agricultural production, and was measured here as the yield increase per unit of magnesium fertilizers applied. The study showed that on average the ergonomic efficiency was 34 kilograms per kilogram, meaning per every kilo of magnesium applied, 34 kilograms of yield increase were gained. Especially responsive to magnesium fertilization were fruit crops, tuber crops, and vegetables. Moreover, there was a positive effect on the sugar content when magnesium fertilizer was applied. So with the application of magnesium, obviously the magnesium concentrations in the leaves could be increased, and this was correlated to an increased sugar content in edible tissues to about 5.5%. So magnesium fertilization is improving the sugar accumulation. The key messages I want to emphasize for this part are: magnesium is highly mobile in the plant and can be transported from all the leaves to growing tissues. It is part of the chlorophyll, the central molecule facilitating photosynthesis, so very important for biomass accumulation of the plant. And large-scale studies illustrated really the positive effect of magnesium fertilization under various field conditions. Specifically, fruits, tubers, and vegetables were very responsive to magnesium fertilization. With this, I want to thank you very much for your attention. I want to give a special thanks to my colleagues Evangelia Papagiannaki, Daniel Nebel, Anna Dutton, Jean-François Sielmann, and Guillaume Touffier for your valuable inputs and your support to make this talk. I want to thank you again for your attention, and now I'm ready to take questions that you may have. Thank you.